Hello, everyone. Welcome. See, a lot of people are connecting. We have around 70 people on the webinar today. Hopefully we'll get more later on. Marie-Noël, over to you. Everything is in place.
Okay. Good morning, everybody. Good morning. I hope good afternoon or good evening, wherever you are. I'm so happy to see you all today again for the second day of our webinar series. So today we will have two presentations on the, mainly it will be on the DNA extraction. So yesterday we had the basic on molecular biology, where we talk about the DNA and the composition of the DNA. Now, today we will see how to extract the DNA from a specific crops. So we have two presenters, Ronald and Verónica. They will bring us through all this protocol. So without further delay, I will give the floor to Ronald, who will start his presentation on the introduction to the DNA extraction. Ronald, you have the floor. Thank you.
Thank you. Okay. Good morning, everyone. I'm going to introduce myself again. I'm Ronald Robles, and I work in the International Potato Center, specifically analyzing the diversity of the collections of wild and cultivated sweet potato a few years ago. Currently, I am making the analysis, the molecular analysis for the collection of sweet potato, looking for duplicates and analyzing the diversity of the entire collection with molecular markers derived from sequencing. And now I'm going to talk with you about the introduction about the DNA extraction, nucleic acid extractions. As a resume of my presentation will be first stressing how fundamental is this process as the first step in all the analysis you will be done with your collections. The second part will be the detail of the fundamentals of the DNA extractions of each process, and also talk about the special cases of molecules or contaminants in the process of the extraction that we need to care. So I will start with a simple example showing how important is the extraction of nucleic acids. For example, in this example, we have an analysis of duplicates. At the left side, we have one cluster. You can see here that imagine after our analysis is done, we obtain a tree. And here we see a group of accessions that are very close together. And we can possibly designate this as duplicates. But also we see another accession in the upper of the group that present a distance that probably make us think that it is different. But what happens if we reanalyze this sample, because we have one doubt, and make another genotyping of these accessions? And the result of this repetition is in the right cluster, as you can see here. And in this image, we see that the repetition sample falls inside the cluster, that the beginning was different. Now we see this accession as a member of this cluster of duplicates. So what happened here? What we can see about these initial results? The conclusion must be that the initial results of the sample, in which the accession shows as a different accession, in reality was the result of the low quality of this initial sample. And this was due of low quality of our sample, our extraction. And the second repetition was the result of a better sample with a good quality. This is one case in which we can see the effect of a low quality DNA. And this example is for duplicate analysis, but also we can see this effect in many other analyses we can make with our collections. So this is one of the reasons why we must to pay attention with the quality of our samples, because this is the initial step in all our posterior analysis. Another effect, apart from what you that you can see here in a direct example of our tree, is that low quality DNA produce poor amplification in the PCR. Another related aspect is that low quality DNA produce a bias in the results, because some alleles are produced at very low frequencies that possibly is not obtained or called in our posterior analysis. And finally, a low quality DNA sample produces low fragments in the digestion. Maybe some contaminants produce that the sample contain some molecules that inhibit the enzymatic reaction in posterior analysis. So for these considerations, we need to have a clear background of how we can make an optimal DNA extraction. Independent from the method, because there are different methods to make the extraction. Some methods are in liquid phase, like the CTAP method and other. And the second group of extraction of DNA is provided by the kits. But in general, the nucleic acid extractions can be separated in some stages. The first one is the sample preparation, in which we need to choose the right tissue, make a good collection, and conserve this tissue in a good condition until the moment of the DNA extraction. The first one. The second is properly the extraction of the nucleic acids. We start with a lysis of the tissue, then the isolation or the separation of the DNA from other contaminants, from the other rest of the cells debris, and this following from a cleaning step in which we make a final removing and separation of the DNA from other molecules that remains in the previous stages. Finally, we obtain a pure DNA that is eluted or maintained in stable conditions for the general purposes. For our cases, we work with gene banks or collections that are composed for many of different crops, species, varieties. We have accessions from cultivated collections and also from wild relatives. And in this variety, give us a lot of diversity, not only in terms of diversity, genetic diversity, but also we faces a different composition of leaves. And this difference make that each species we work is a very different world with we want to work. For example, some species contain more polysaccharides than others. Other species don't produce a lot of polysaccharides, but contain phenols, polyphenols, and other metabolites that can affect the same process of the DNA extraction, and also can affect the posterior analysis of our samples. For these reasons, the sample preparation, the first step, is a key factor for the assuring a good quality of our DNA extraction. This step, the sample preparation, can be divided in two moments. The first one is the selection of the tissue. Here we need to choose the right tissue in which we will obtain a better quality of DNA. Mostly are recommended the young leaves because the young leaves have low amounts of secondary metabolites and is better for the obtaining good quality DNA. But you can also work with mature leaves, with root, even seeds, stems. It's better to, when you starting a project with one species, you be assured and try to make this first step to analyze and look in which tissue is the better for you. And the second moment is related when how you will storage and maintain the collected tissue from the field, from the greenhouse, or maybe you are making a collection in another area, and how you will be storage and preserve that collected tissue until the moment in which you will be done the DNA extraction. In this second moment, you need to care how you will preserve the tissue. If the tissue will be maintained fresh, or it will be stored in a buffer or frozen, or you will desiccate by many methods available. Here I have a table for the first consideration, which tissue you need. Here we have the results of the amounts of DNA that are produced by different tissues. Here we have the young leaves. They also tried to extract from matured leaves, seeds, roots, and the stems. And you can see here that the better results, the highest amount of DNA is obtained by leaves. The amount of DNA is similar between young and mature leaves, but in the next slide, we will see that not only the amount of DNA is important, but also the quality. So with respect to other tissues, the leaves are the better options. And from this, the young leaves are the optimal. In this another table, we will see that not only the DNA amount is what we need to look, because here we have a comparison of the amount of DNA that was produced by leaves and from roots. And the first results, we see here that yes, the leaves produced more amounts of DNA, 3.2, it's a mean, versus 2.1 in the root derived tissues. But in the another rows, we see here that, for example, for the leaves tissue, maybe almost all the sequences that was obtained in a sequencing analysis of this sample, almost all of the DNA sequences was belong to the species. 1.02 sequences reads produced, and also 1.02 reads belonging to the species. But in contrast, the amount of DNA that was obtained from the root tissue, from the 1.55 millions of reads that was obtained, only 0.61 belonged to our species. That means that we cannot rely only in the amount of DNA we obtain. We have to consider that depending on the tissue we choose, the quality was be considered by the amount of the DNA, and also considering that some tissues produce contamination. In this case, the roots, the amount of DNA that was obtained from the roots was not only from the species, but also from another microorganism in the associated in the tissue. The next consideration is the storage. Suppose that we find and choose the right tissue and start our collection. Now we have to have a plan to establish a methodology on how we will store the tissue that we collect. I mentioned previously that in some occasions we have a greenhouse or a field near to our laboratory. In this case, the logistic is not so complicated, but in cases in which we have to collect samples that are at long distances from ours, we need to plan a method to preserve the samples we are collected. One option is maintain the fresh tissue. Even in long trips, we can maintain the long tissue, for example, by putting the lips or the tissue in towel papers, by moisturizing the towel papers. And this procedure maintains the tissue in fresh conditions. But in the graphics that we see here, we can see that we need to be careful to not to preserve the conditions of the samples. The better strategy is to collect the samples, the tissue, and store it in liquid nitrogen. This is due because at the moment in which the sample is collected, it starts to degrade, and some cells are release their content. And we need to avoid that the content of the cells is released because this starts to damage our DNA. For this reason, another approach consists in store our tissue directly in the extraction buffer we use. This must be done with careful because, as I mentioned, at the very moment in which we collect the samples, it starts to degrade. So another method is to desiccate the sample, remove all the water as possible from the sample, and very quickly to avoid the degradation of the tissue, the cells. Here I have one analysis that was done considering many options that are used currently. For the method of the storage, for example, they tried to store the leaves, the tissue, directly in ethanol to avoid the degradation of the samples. Another option was using isopropanol, another alcohol. And the third one was to store the tissue in a mixed solution of chlorodium sodium and CTAP, is a detergent. And this three was compared with another three methods of storage in which the samples were desiccated. Could be in silica gel or only put the leaves in tower papers. And finally, the sixth condition was to store our samples in low temperatures. Here they use dry ice because the liquid nitrogen was very difficult to make this, but it corresponds. And after applying the same DNA extraction protocol for this method of the storage of the tissue, they look what was the results, the quality of the DNA extracted. And we can see the first methods in which they were the tissue was obtained and preserved only in paper towel or desiccated in silica gels or frozen in low temperatures, as in the case of dry ice or liquid nitrogen. These methods provided the best results for the quality, and the lowest quality was obtained when the tissue was preserved in alcohols or in another solution of CTAB or sodium chloride. But even in the lower image here, in the lower group that you can see here, isopropanol, CTAB, chloride sodium, we can see that some species produce better and good results for these methods. For example, we can see here that the species A and B produce good results when other species fails in this method. So this tells us that we need to prove. We cannot make the error of planning a project and choose our method of tissue collection and preserving of the tissue based on a recommendation or based on our previous studies, and not trying and not see this, make these initial screenings to confirm that our species, the species in which we works well with the recommended conditions. The next steps in which the DNA extraction will be see in detail is the lysis, the lysis of the tissue we are collecting. In this step, the main objective is to disintegrate all the cells. The vegetal cells have not only the cellular membranes, but also another structures in which the tissue is very difficult to grind, to break. So for this reason, we use some molecules that will be help to degrade the tissue and release the content, because we need to release the DNA from their nucleus. To help this lysis, we make mechanical maceration. We can opt to grind the tissue first in mortars or put ceramic bits inside the tubes in which we will make the DNA extraction. And the result will be that all the compounds of the internal organelles in the cells will be released and mixed together. In a living organism, the DNA is preserved inside compartments of the cellular. And for this reason, our DNA is well preserved and functional. But at the moment in which we make this mechanical maceration, all the compounds of the cell are mixed together, and the DNA is in this moment is prone to be degraded by the other compounds of the cell. This is the reason why we need to take special careful from avoid the degradation of our DNA. The compounds in this stage are the CTAP, which is one of the main compounds for the DNA extraction. It's a cationic detergent, and their mechanisms of action is to degrade the membranes of the cell, solubilize the proteins to avoid they interact with the DNA. The other compound is the tris, and in a pH of 8, because this molecule acts as a buffer to maintain a stable pH to avoid the degradation of the DNA, and also to avoid the activation of another enzymes that was released from the cells. The third compound is EDTA. This one have the property of capture ions of the mixing cells. In this compound is very important because their capacity to bind molecular cations permits that some of the enzymes start to be inactivated, because the enzymes that mostly degrade the DNA needs as a compound to be activated some ions as magnesium ions or calcium. So the EDTA act capturing these ions and avoiding to the enzymes to be active. The fourth compound is the sodium chloride. It's very important because depending of the concentration, if it's we make a lysis buffer with high sodium concentration or lower sodium concentration, we will be obtaining different results. The normal protocol for the lysis of the in the DNA extraction use an amount of sodium chloride that promotes the precipitation of polysaccharides. In cases in which we have more amounts of polysaccharides, we can increase these amounts, the concentration of the salts, to avoid that the polysaccharides interact with DNA. With higher concentration of sodium chloride, the CTAP is binded with the polysaccharides and not with the DNA. And as I've mentioned previously, there are some secondary metabolites that some accessions, some species present. There are the polyphenols and the polysaccharides. And to avoid these molecules oxidize and bind to the DNA, we added 2 additional molecules. This the beta-mercaptoethanol. And this molecule have the property to inactivate proteins because they cut the bonds of the enzymes and denatures the structure of the proteins, being inactivating some enzymes that can damage our DNA. And the other is the PVP. This is the polyvinyl pyrrolidone. This is a large compound, molecular of bigger size, that act as act by attaching and binding and sequestering the compounds like polyphenols and polysaccharides to avoid the interaction with our DNA. So these molecules in combination give us also the lysis of the membrane, the release of the compounds, and also avoiding that any other enzyme or molecule can degrade our DNA or inactivate. Now we go into the next step. Here is the isolation of the DNA. This mix of compounds must be separated, and we obtain this by combining with a molecule of chloroform, the principal solvent here is the chloroform. The action is to solubilize the proteins and another component, organic compounds, to remove this from the DNA. And because of their higher weight, after a cycle of centrifuge, the DNA will be appear in the upper phase in combination with the CTAP at this moment, and the chloroform will bind to another compounds with another organic compounds and will appears in the lower phase. This will make permit us to separate the DNA from the rest of the debris from the cell. In this step, we need to make gentle vortexing, but not so much to avoid the DNA fragments will be shared. And one of the very key stages is the cleaning of our elution. Here we have 3 or 4 compounds that are used for the cleaning of our DNA. One is the isopropanol. This is another alcohol that can be used to precipitate. In this step, the binding of the DNA and CTAP will be released because isopropanol is a polar molecule. So the addition of this molecule will break the bonding union of the CTAP and the DNA. The result is the DNA will precipitate, and to help this precipitation, we use this isopropanol in lower temperatures to accelerate this precipitation of DNA. But one consideration is that the isopropanol at lower temperatures can also co-precipitate phenols or other compounds that still remain in our phase. So another option is to make this precipitation at room temperature, but the times required to the DNA precipitation at room temperatures increase. We can take an overnight precipitation, for example, in the case in which our tissue is with a lot of contaminants, for example, with phenols. After the precipitation, we need to erase any amounts of salts or any amounts of CTAPs from our pellet. And for this, we use ethanol. We make the washes with alcohols. Can be done at room temperature or with lower temperatures. And in some cases, it's better to combine this washing of alcohol, adding some salts. You can use sodium chloride salts or sodium acetate salts for cases in which polysaccharides still remains in our final pellet. And finally, at the moment in which we obtained the most of the pure DNA possible, we make the washes with more alcohol to remove any other residuals, and finally obtain a pure pellet of DNA. This pellet of DNA is finally resuspended, and you have here another options. One is to resuspend directly in water, but this can is not good for a long term of preservation of the DNA. So we use this in case all the DNA will be used in short periods. But if you want to conserve the DNA for longer periods, we use specific buffers. Here, the most common is the TRIS EDTA buffer, in which the EDTA will act also to avoiding that any contaminant will degrade the DNA. So the pH also will help to maintain it stable, the DNA, for long periods of time. In combination, these steps are the basics from the DNA extraction. And as I mentioned earlier, you in the next presentation, we will hear a lot of protocols and specific conditions in the protocol that you will be use to your specific tissue or your specific condition. But at the very beginning, what we need to do is not rely in a specific protocol for our sample, but also which are the samples, how the samples will be chosen, and how the samples will be stored and maintained until the moment of the DNA extraction. These initial steps are very fundamental to the obtaining a good quality of the DNA. So for the moment, this is the steps from the DNA extraction, and I will pass to Verónica for the next chapter.
Hello.
Thank you very much, Ronald, for the very interesting presentation. So Veronica, you have the floor now for the second presentation on the protocol. Thank you.
Yes. Okay, good day to everybody. So first of all, I would start talking a little bit about me. My name is Verónica Valencia Límaco. I am a biologist with a specialization in botany, and I am currently studying for a master's degree in bioinformatics and omics sciences. I am a research assistant at International Potato Center's Genebank, and I have both experience in wet lab and in dry lab. So I've done a lot of DNA and RNA extractions and PCRs, but I also do bioinformatics and general data analysis. My main experience is working with underutilized Andean root and tuber crops. So tubers you've never heard of, probably. And I do a lot of molecular characterization for conservation strategies. So I'm going to talk about here an overview of DNA extraction protocols. So I would start with advantages and disadvantages of two main types of DNA extraction methods that are setup and commercial kits. And I would talk more deeply about the setup method and optimizations we've done here at SIP and other common modifications. And I would end with good practices. So the first thing we need to have in mind, and it's something Ronald also said, is that the DNA extraction is the critical first step in any genetic analysis, and choosing the right method is a key to success. So even if we have very modern sequencing techniques, everything starts with a good DNA extraction, and it's something we have to be very careful of. So we have two main types of DNA extraction methods. There are others, of course, but a classical protocol is the setup method that Ronald already explained the basis of, and commercial kits. But they aim for the same, that is, the break open plant cells, remove contaminants, and recover clean, high-quality DNA suitable for downstream applications, like, for instance, sequencing or PCR. So the setup, the classic protocol, the first advantage is that it's very cost effective. because it uses inexpensive and widely available chemicals, and is ideal for large scale, or if you have a low budget, it's always good to have reagents to do setup. The other advantage is that very flexible and customizable. So because you can adjust almost any step of the setup method, and you can customize it to the plant you are working with. The other thing is that it produces a high DNA yield in comparison with commercial kits. You can have 1,000 microliters per nanogram very easy, and also is effective for what we would call difficult plants. It's like you have many contaminants, you can adjust the method for those contaminants. And the other thing is that it doesn't have proprietary components. Many kits have, they don't share how their kits work, and you cannot reproduce it. So it's also an advantage that they don't have proprietary components. The disadvantage is it's very time consuming. It takes time. We, for instance, start very early in the morning and we don't finish it till the end of the day. So we have to be in the lab all day, and requires hazardous chemicals like chloroform that it can burn, and even you cannot even smell the chloroform. So it's a hazardous chemical. It requires technical skill. You have to depend on user expertise, and you have to practice a lot for the protocol to work correctly. And it's, again, labor intensive. And you can still have, even if you modify and optimize, start optimizing your setup method, it produces some issues so that can cause that your PCR doesn't work well, or the sequence it's not up to the standard of the company you want to send your DNA. So in contrast, we have the commercial kits. The advantages and the big advantage is that it's very convenient and it's quite fast. The other thing is that it's reproducible and it's consistent. So every time you do it, it's going to produce the same results. And it's quite safe. It doesn't use many hazardous chemicals. And produce high quality and high pure DNA, right? And it's usually free of inhibitors, so it's very ready to use for sequencing. And many companies recommend that you use a commercial kit for some platforms of sequencing. The great disadvantage is that it's quite expensive and may yield produce poor results with challenging blood samples. In the use of a commercial kit, compared to setup, you can have maybe 20 or 30 nanograms per microliter. And it's especially when you have challenging plant samples. You depend a lot on suppliers too. I've had a case where I asked for a specific commercial kit and it took about 4 months to arrive. So that's also a great disadvantage. So the other is that it's not that flexible to optimize. Normally, commercial kits are made specifically to solve some problems, right? So it's not a setup that you can add or take things. You can do some modifications, but it's not that flexible. The other thing that I already mentioned is the intellectual property and transparency, because we sometimes don't know, like for instance, with dilution buffer, right? We don't know is nuclease-free water, is 3-CTTA, is another thing. So sometimes they don't specify what are their reagents. And the same sample size limitations. The kits come to you can have a kit for 5 samples, for 50 samples, 100 or 200. So you have to plan for that many samples, right? So those are the advantages and disadvantages. So when we talk about, like we pass to talk about the optimization. Of course, we have to talk about the great protocol that is, I think, in most articles you would read, right? That is Doyle and Doyle. This is one of them that is mentioned. That is the article from 1987, a rapid DNA isolation procedure for small quantities of fresh leaf tissue. But the canonical protocol is this protocol from 1990, isolation of plant DNA from fresh tissue. That is a refined version for broader plant tissues and more widely cited in modern protocols. And this is the one they are referring to when in your article you read modify from Doyle and Doyle or following Doyle with modification. So even as when we report DNA instructions, we say modify from Doyle and Doyle, and you probably would do the same. So it would be worth to read. Okay, so to keep going, you have to keep in mind that for CTAP, there is no one size fits all protocol. There is no protocol that works in everything, in every species, in every type of plant or tissue. So the best approach, if you want to start doing DNA extraction, is that as Ronald said, it depends on many things, like the crop type, but you also have to take into consideration the available resources you have. Not every lab has every equipment, and also the technical expertise of your team. So adapting protocols to your local condition is essential for sustainable genetic conservation. So starting there, we need to remind ourselves what are the 5 main steps of a DNA extraction. We have the sample preparation, the lysis. After that, we have isolation, the cleaning, and dilution. Right? So this, I don't know if you can see it. It's a little, it's tiny, but I put a table. In the first part, I am putting how is this process in Doyle and Doyle 1990, and how we exceed modify it for, this is an example for a sweet potato. So for instance, you have the sample amount for when your for sample preparation. Doyle and Doyle says 0.5 grams of young leaf tissue, and we use up to 0.22 grams. So it's less than half, because we found out that with this amount of tissue, after doing some tests, that we produce better and good quality DNA than with more tissue. Not always more tissue is more yield, and not always is high quality. So in lysis, the main modification, the main optimization we did was that for Doyle and Doyle, you would use liquid nitrogen, mortar and pestle, and we do liquid nitrogen, but we mostly do ground with ceramic beads, and we do it in an equipment called fast prep. So that is another thing. The other thing is that in Doyle and Doyle, for instance, they use 700 microliters of CTAP buffer and 0.2% of beta-mercaptoethanol. We do maintain this proportion of beta-mercaptoethanol, but we use less CTAP right? So this is a modification you would see a lot in many protocols. The quantities of what you add sometimes it's a little bit more, and sometimes it's a little bit less. The other little but the other little optimizations we do is in time. The incubation from doyle is 60 minutes, and we do a little bit less, like 45 minutes, and we mix in every 10 or 15 minutes we mix. After that, another key difference would be that we do 2 chloroform phases to improve polysaccharide removal, and we do 2 washes, right? Not only one with 70% ethanol, but we do one with 70 and another one with 90% ethanol. And for RNA removal, we do a long incubation of 2 hours instead of 30 minutes. So this would be our optimization here for sweet potato. So you can start always with Doyle and Doyle if you want to try it, but it's very common to have 2 washes and 2 chloroform phases. Okay, so I put here some pictures of how we have our materials. We collect samples from the field, right? You can see the field. I don't know if. Okay. So we collect samples from field, right? So we go to the field and collect, but we also have greenhouses. So we sometimes collect from greenhouses. And since we work at the Genebank, here at the Genebank, we have in vitro material. So as Ronald said, we mostly use and prefer young and fresh leaves because they produce normally high DNA yield and they have fewer inhibitors. So here you have a picture of how would we have the material collected in CETA. We normally use tweezers, but it has to be sterile tweezers. And if you want to pass to another accession, another sample, you have to re-sterilize the tweezers. We do it mainly with alcohol. And we make sure that there is no rest of other plants, etc., on the tweezers. So when we talk about the lysis and grinding techniques, one of the most common is probably the grinding with liquid nitrogen. Liquid nitrogen prevents the DNA degradation because of the temperature and facilitates fine powdering. So when you have a very hard tissues, it can solve many problems. It does have a disadvantage that is that not everybody has access to liquid nitrogen, and it has to be managed with care. Right. The other thing is that you have to do this mortar, and so you have to have lots of this if you want to grind faster. If not, you have to clean it, and then clean it and clean it again, right? But if you don't have liquid nitrogen, a protocol I've used before and has worked very well is that you can use polypropylene or polyethylene bags, right? And you can put your tissue here, your leaves here, and you can with this same I don't know how to say, I forgot the name. You can grind by hand, right? I use even a tube from in vitro that is clean and empty, and you grind, and then you add your setup, and with a lot of care, you put it on a microcentrifuge tube, right, without touching anything in the surface. And this we do mainly for RNA extractions, but it's a good option if you don't have liquid nitrogen and if you don't have a fast prep. It could be a solution. But what we do, this is part of our normal functioning, is that we use this machine called fast prep. As you can see here, the material we collected, we add this pellet, this ceramic bit, sorry, for each one, and we put it in the machine. And okay, I put here a video. Sorry. So we normally do 2 cycles of 20 seconds, right? So this machine works for us, so we don't have to do it by hand. Now there is some companies that recommends us to use their own proprietary bits. Sometimes they are not reusable, so sometimes they come with kits, etc. But for setup, we use our reusable bits since it's cheaper for us, right? So this is how we do the lysis. Okay, let me pass to. Okay, isolation is probably the longest step. That is not a step, that is many steps. So after we grind the tissue with the fast prep, we do an incubation at 65 grades Celsius for 45 minutes, as I said, in this Baño Maria, I don't know how to say, Maria bath. So after that, we take it from this machine and we let it cool off because it's hot here. After a little bit of cooling off, we add the chloroform isoamyl alcohol and we put it in the centrifuge. And this is the important part, that we have a tube here with the phases. So the ceramic bit goes to the bottom. Then you have the organic phase and the interface with the chloroform and the rest of the tissue. And here our DNA is with the setup in the aqueous phase. So the aqueous phase is the one we're interested in. And we have to be very careful not to touch the interface of organic phase when we do the supernatant transfer. So we use whiteboard tips for this. And even if you leave a little bit of the setup here, it's better than to contaminate your sample with chloroform or a little bit of the organic phase of the interface, right? So we passed from one tube to another clean sterile tube. And after that, we add the setup. But this setup is preheated and it's more concentrated. It's a 10x, not a 2. So we normally use about 70 or 80 microliters, right? And after that, we add chloroform, we centrifuge, and we have another phase separation after the centrifugation. But as you can see, it's cleaner. So that's why we repeat chloroform to have it cleaner, right? So after that, we do another supernatant transfer, because we don't need the chloroform here. So we need another supernatant transfer, and this we add the isopropanol. We leave it 30 minutes at -20, and then we remove the supernatant, right? So we can isolate the DNA pellet. Okay, so the cleaning phase, I had to put here just these images because I couldn't find the pictures. But we have here two cleaning phases. We add chilled 70% ethanol, and we invert the tubes to wash it, right? We remove the supernatant, we add chilled 90% ethanol. We again do the same, invert tubes, and remove the supernatant, and we let it dry. We let it dry upside down about a minute or two. But when it's the when we're finishing, we leave it from one day to the from overnight with cover and just just a little hole in a saran wrap, so this can keep dry. But it's not necessary if you see that the DNA is, sorry, I didn't change this. If the DNA is already dry, and after that we add water, nucleus freeze water on NFW or tris EDTA, right? So we have our DNA pellet. This is a part of the elution, and we add our 3C DTA. And you can see here the pellet. We add this water or TA, and you can see how it starts resuspending, right? So that would be all for how we do the DNA extraction here at SIP. But I would recommend this article that Ronald and I based off to talk about some modifications here that questions why everybody puts modify setup protocol, but they don't specify how they modify the protocols, and it's not why they are not reproducible. It's a good article. What I liked the most was the other common modifications, like adding mercaptoethanol or polyvinyl or proteinase K, RNase A, etc. So everything has a function, like Ronald already explained, but I would like to talk about my experience. I work with Andean root and tuber crops, so they are quite different. We have, for instance, Uyucus tuberosus. That is a plant that has a lot of mucilage in the leaves. It's a lot of gum. And while the kit that we use works very well, it becomes stuck because the gum doesn't let the filters work. So we did a modification with sorbitol that you use a sorbitol wash buffer that is a pre-treatment before you enter the CETA or you enter the protocol for the kit, and it works very well. But for instance, another plant, the Tropaeolum tuberosa, that is another plant I work with, that it has also gum, it has mucilage, but it's very little. So in this case, washing it with sorbitol doesn't help because it's so little that it doesn't affect the quality at the end. And when you wash with sorbitol, you take a bit of the yield, so it's less concentrated at the end. So we have to be very careful about doing that kind of modification. Another thing, another plant I use, I work with is Oxalis tuberosa. And in this case, the recommendation I saw a lot for these contaminants of oxalates, these salts, was to add more polyvinyl So we made a modification with polyvinyl and also acetate because we wanted more clean, but it didn't work at the end. It helps a little, but we didn't manage to standardize set so to have great quality. It was just good enough, close to good enough. So the kit for oxalis works very well because it has a cleaning step that that works very well. But since sometimes we don't have kit, what we find out is that if you use the sprout tips instead of leaves, you don't have any problems with oxalates from OCA. So that these kinds of modifications help a lot with setup. And so that would be examples of my experience. The other thing with mercaptoethanol is that sometimes it's worth adding a little bit more, especially if you are maintaining the sample on the freezer and you are not extracting the same day you collect. If it's not the same day, sometimes you would want to add more mercaptoethanol to prevent oxidation. And I made a modification too for the kit that I was using a little bit of mercaptoethanol because it helped me to recover more yield, right? But you have to be very careful with mercaptoethanol. It's a common practice to collect in setup with mercaptoethanol, but we have to be very careful not to inhale because it smells it smells horrible, but it's quite dangerous. We have to be very, very careful. So beyond the standard good practice to pass to good practices, beyond the standard good practices like wearing gloves or cleaning the surfaces or avoiding contact of pipette tips surfaces, there are quite common recommendations of any molecular laboratory. And I think that anyone who is working in a molecular laboratory will have those good practices. For specific precautions of DNA extractions, use only sterile if you can and DNA-free consumables. Change tips frequently and to prevent cross-contamination, and always label your tubes. If you can, have a notebook or a lab notebook and always write down what you're doing that day, because you would know that day, but maybe in 2 weeks you won't know, right? But even beyond this, I would like to give some personal recommendations for DNA extraction based on my experience working with people doing their thesis and interns or new research assistants, etc. Is that if you decided to work with a protocol, even if it's a kit protocol of a published protocol, you do it exactly as written before you do any modification. Sometimes we jump out to do the modification, and so if it works, if it's not broken, don't fix it, right? So do it the first time you do it exactly as written. So if modifications are needed, don't do 4 modifications at a time. Change if you can only one factor and identify what's its effect, right? And the other thing that is very important is that you begin with a small scale. If you're starting to extract DNA, maybe 4 or 8 samples, depending how much you can sacrifice before scaling up. Because I've seen this problem a lot, especially with university students that are doing their thesis, that they have 100 samples and they want to do the 100 samples. So they upscale from the beginning, and they after that they have to repeat everything because they forgot to I don't know, add mercaptoethanol to their seed, so they lost the 100 samples. So always start, especially if you're working with a new protocol, with just a little. And this is a call for any supervisor that please give people at the lab, and if you're in the lab, request your supervisor that they give you time for protocol standardization and troubleshooting, because sometimes they want the new person, the new technician, to do these protocols in a week, right? But that is how we end up with bad quality DNA. That's how we end up not being able to work with the information that from sequencing and the problems that arrive in the bioinformatics, the bioinformatic analysis, etc. Right. So be very conscious and have time to do any protocol standardization. Thank you. That would be all.
Thank you, Veronica. Thank you so much. I see we already have a hand up, so I will give the floor to this participant. We have Otman. You raised your hand. Now it's down again. Perhaps Ali? Ali, you can unmute yourself.
Did you hear me?
Yes.
Okay. Thank you for the clear two presentation. Are very fruitful for me, and I have only two questions. The first one regarding the sampling methodology. As you know, in the our seed bank, the local variety and local accessions have many accessions or many lines in the same accession. How to can do the sampling in this way? Each line alone, or we mixed together to analyze the DNA and the PCR protocol. And the second question, if I collect my DNA and I have a bad quality of the DNA, I can improve it, or I would take, or I would restart the protocol again. This is my two questions. Thank you.
Okay. Ronald, you want to answer, or me, about sampling?
Yes. About the sampling, you're referring about the sampling strategy of collecting from many samples in one extraction. I understand that. And that depends of your approach, because one option is to extract the DNA from each sample, from each individual, when you have to, when you want to analyze the diversity of the intra-population. In our case, we work with clonal collections. For this reason, we only make extraction of one clonal sample and represent the entire accession. But there are also crops in which they conserve as population of seeds, for example. And in that cases, you have to take care of the consideration. If you want to extract as a batch, a bulk of samples to represent the diversity of your accession. In this occasion, in these scenarios, is avoidable that you can make the extraction in one batch, and only if you will be analyzed as a bulk.
Mm-hmm. Thank you.
And
yes.
the other,
yes.
yes, Veronica.
No, go ahead. Go ahead.
The other question was about how you treat your extraction protocol when they fail. I understand that.
Yes, my second question is how to improve the quality of my collected DNA.
Yes, and that's what I mentioned at the beginning, that previous to looking at a better protocol or standardize a protocol according to your samples and your conditions, it's better to standardize the process of collecting the samples and to preserve the material. As you see in our presentations, we presented that there are many trials that you can make for choosing the tissue or the method of the conservation. One of the most easy ways to achieve better results is to lyophilize the samples. That is in the case in which you have a long distance to the collection site to your laboratory. If you have very easy the access of the samples to your laboratory, the normal is to work with the fresh tissue and make the extractions at the moment. But the first step is to consider the better choice in your sample preparation. If you have access to nitrogen, liquid nitrogen, it is the most better option. And the other is the case of our next session tomorrow, in which we will address a specific challenge of the extraction, because in our laboratories we don't have all the equipment for all the accessions, for all the tissues. So we have to combine some challenge for this is we will talk about tomorrow.
Yes,
Yes, so
thank
just
you.
a compliment. Just as a compliment, I think that sometimes it happened to me that I isolate DNA and it was not the best quality, and sometimes the sample is unique and it becomes a problem. So there is some protocols for resuspending and cleaning DNA, but it comes with a risk. If it's not that concentrated, you would lose a lot of yield doing this cleaning after DNA.
Yes.
And
Yes.
if you can, maybe you can restart. You can do. I think we do normally is that we do a sample and it's copied. So we have one to save us if the other one fails. So you can maybe explore a bit about this cleaning afterwards. I think that you can resuspend with acetate, etc. But for me, for the case I have with this difficult plant of oxalis, even if I did all of it and it didn't work, so I had to restart at the end.
Sorry, can you send us the cleaning protocol for the DNA?
I think I can find it to resuspend. Yeah, there are kits to do that now. Concentrators. Stuff. Yeah, no problem.
Thank you. Thank you.
Thank you so much, speakers, for your answers, and thank you so much, participant, for your question. We have loads of hands up, so I will just move on with the next. Jantor Ndalo, you can speak whenever you want.
Thank you so much for the insightful presentation, and I have learned a lot. I would, however, would like to seek your indulgence, Valencia, with regards to isolating DNA with high mucilage. Sometimes in the process of isolating, you find DNA has entangled around the mucilage. What do you recommend? That was number one. What are the recommendations from your side with regards to some of the modifications we can do on the CTAB method? And then number two is with regards to high molecular DNA isolation, what modifications can we do on CTAB isolation method to have high molecular weight DNA? Thank you so much.
Okay, so about mucilage, as I already talked about this. I have two plants that have mucilage, right? So Oyukos and Tropaeol. So Oyukos have a lot of mucilage, a lot of gum. So even when I was working with kids, we have this problem, right? And when I did CTAP, the end, when I do the elution, we have gum with DNA, right? But in the case of Mashua, it was not that much gum, that much mucilage, so it didn't cause any problems. The modifications I did was prepare a sorbitol buffer. It is a protocol before you do the CTAP method. So you prepare this wash protocol, this buffer. It's a buffer. So you do basically you grind with the sorbitol buffer, and after that you centrifuge and you take when you discard the supernatant, the gum goes with the sorbitol, and you have your leaves not completely clean, but without many mucilage, and you can start your CTAP method. But it does produce some problems if you are not technically skilled. You can you can leave much sorbitol in it, and if you would have at the end not that very good DNA. So you have to be very careful with the washing, right? This is a protocol, I think it's published, if I'm not mistaken, the sorbitol wash buffer. If not, I think I can send the one we've prepared so you can try it. Yeah,
Sure,
but
I'll
you do
really
sacrifice.
appreciate it.
Yeah, you do sacrifice yield because it's you're taking away some of the
DNA.
sample. Yes.
Okay.
Yeah. What was the
I
other
think
one? To high yield.
Yeah, high molecular weight DNA. What
Correct.
modifications can we make to achieve that?
High molecular weight.
For the you have to want to use this DNA for high throughput sequencing, maybe.
Exactly, for long read sequencing.
Yes.
Yes.
In some occasions, the CTAP methods work well for SNPs genotyping with sequencing of the DART6 or GBS. The CTAP method works well, but we have another occasions in which we want to make the PAC-bio sequencing. And in that, for that platforms, the CTAP in some species don't work well. What you could try is to make your CTAP method, but with lower temperatures, maybe in the incubation, and also
the CTAP buffer.
The CTAP buffer, the incubation in the water bath.
Okay.
To avoid the degradation of the DNA, reduce the temperature of your incubation, and also trying to
What is your
make
recommended reduction in temperature?
Yes, reduction of the temperature.
Normally
in the
we
incubation.
use 65 degrees.
65 is the normal, yes, but you can lower the temperature 10 grades, no more, to increase the amount of the good quality of the yield. And also, I don't know how many tissue you use normally, but sometimes when we reduce the amount of tissue, the proportion of tissue and buffer, in some cases is do better results. We obtain less DNA, but with more quality. And this is one aspect we also want to take about is the how much tissue we use in relation to the volume of the buffer. And this is one of the main considerations. Maybe you look in your results, your yields, agar yields, we can see specifically what condition we can choose.
Okay, well, great.
Mhm.
Thank you.
Yes.
Thank you so much.
Thank you, Jantor. I hope that answered your question. We
Yes,
have
I
loads of
answered.
questions, so we'll try to go through them all. But if we don't manage, I'm sure Veronica and Ronald can share their contact and you can reach out to them
Yes.
directly. So we have a question from Ahmad Shakil.
Well, thank you very much. Can you hear me?
Yes.
Am I audible?
Yes.
Yeah.
Oh, okay. Okay, thank you very much. First of all, thank you very much for such a wonderful presentations and such a rich in knowledge content. I have a couple of questions. Though it's a very basic molecular biology experimentation, but for my own understanding, do you think there is any optimum ratio, as we all agreed on that, okay, liquid nitrogen is the best way to grind the tissues to lyse the cell. Okay, so I want to know, my first question is actually, I want to know whether there is any optimum ratio for the usage of liquid nitrogen with respect to the tissue or the time. How much time we should grind, how much quantity we should use. For example, we have 2 grams of leaf or 3 grams of leaf. Okay, so what quantity, for example, is there any proportion roughly we can give? For example, if we are using 10 ml, 30 ml, 20 ml liquid nitrogen. So the first question is this: is there any optimum ratio, or is there any effect on the DNA integrity if we are using high amount of liquid nitrogen for grinding, or we are grinding our tissues for a long time using liquid nitrogen? So what will be the effect on the DNA integrity? So can I go for the second question, or after the answering of this question, can I answer the second question? What do you suggest? Should I ask the second question,
I
or
let
should
the
I wait for the...
speakers. I'll let the speakers decide.
We can respond now. Yes.
Okay, go ahead.
Veronica, you or you want to respond? Because no? Okay. For the amount of liquid nitrogen, there is no specific ratio, but there is you have to consider which amount you need to a proper grinding of your tissue. This is the key. How much you need to obtain a good grinding of your tissue, because it depends of your tissue, the leaves, how much thin are the leaves, but you will be known how much because you know how much time takes you to obtain a good grinding of your samples. And if you see that there is a lot, there is too much, you need more time to grind, and the samples lose the frozen of the samples. You need more. And the other thing is that when you make the grinding in liquid nitrogen, you don't need too much time to put this tissue in the buffer. The most optimal is that if you have small times, a quick time to make this, you add the extraction buffer directly in the mortar pestle to break the action of the metabolic compounds. For this reason, you in resume you need the amount of liquid nitrogen exactly in which you need the time for the grinding. if you add more, there will not be another effect on your sample. There's no specific effect of much or less.
Oh, thank you very much. So you mean excessive use of liquid nitrogen is permissible, and there will be no negative effect on the DNA integrity if we are using somehow. Okay,
Yes,
all right. Thank you very much. Thank you very much for clearing this point. My second question is related to the tissue as well. Do you think there could be any chilling effect on the DNA yield? If, for example, our samples, we are directly tissue samples actually have been directly exposed to some ice or some chilling conditions, and there are some dead tissues, so they can that injury can affect the ultimate DNA yield, quantity, quality, anything.
Yes, the optimal, if you have access to ice, dry ice, liquid nitrogen, or an environment in which your samples are in low temperature, is the optimal. But you have to consider that if you are providing this environment to your samples, you must be very careful to not break this frozen change in your methodology. So yes, the chilling of the samples help to avoid the degradation of your DNA, but you have to use this properly, because at the moment in which you are started the lysis of the sample, you need to add the lysis buffer and the other components like the percaptoethanol and PVP. This to help your previous treatment of your samples.
Thanks, Ronald. It's good. Now I have only two questions. Post DNA extraction. Excuse me. Do you think there could be any freezing and thawing effect on the DNA quality? Say, for example, we have extracted the DNA and know whether to kept at 4 degree or minus 20 degree or at room temperature. So what do you think? What will be the life of the DNA? I know, okay, as minimum as the temperature will be, okay, the life will be the more. So my question is related to freezing and thawing. Okay, we are taking sample out from the refrigerator, we are working on them, we are taking them back, putting them back into the refrigerator again, we are working on them, and we are not using, for example, if we are not using the ice packs, we are not using the ice flakes, okay, neither we are using dry ice or anything else. So we are just working we are just directly working with the DNA, and okay, there is a freezing and thawing. Okay, our samples, DNA samples were initially freezed, later on, okay, we were working on them, any sort of working, we were labeling them, we were sorting them, anything. So do you think there could be any effect of freezing and thawing on the DNA quality or quantity?
You're referring to your DNA already extracted, correct?
Yep.
Yes. Yes. As you referred, the continuum using of your samples to the frozen state, to as you mentioned, you put your samples and they go back another time. This also reduce the lifetime of your DNA. This is a fact. The optimal is that if you are going to do these procedures, separate aliquots of your DNA from the rest of the obtained DNA. For example, after your extraction, and the moment in which you elute your samples and store it, in that moment you make aliquots of your DNA to avoid this use of the same tube from inside and outside of your refrigerator. And also you need to make a constant analysis of the quality of your DNA if you don't have the option to make the liquids, and you sequentially make the agarose gels of your DNAs to be sure that your DNA is still in good conditions. That's what my recommendation.
Perfect, Ronald. Thank you so much. My last question is related to transportation issue. Okay, for example, if we are shipping our DNA samples, we are not lyophilizing them. We are not using dry ice. Okay, so what are the appropriate ways to ship our samples, for example, to outside the country or somewhere at a long distance?
Yes, we in SIP make the genotyping of our collection from potato, sweet potato, and Andean rhubarb and crops, and we send the DNAs from Peru to Australia. It's a very long trip that our samples make, and we have established a protocol for the delivery of our samples. We use, as maybe you can use also polypropylene or maybe styrofoam boxes. And our samples are also the DNA in resuspended condition. We add to the box gel pack at the moment in which make the package of our samples. We add 1 or 2 gel packs inside the box, and this gel packs works well for all the trips for 3 or 4 days of distance from Peru to Australia. And in some occasions, the customs of Australia retain our package, and the total amount of time was more than 1 week, and our samples resisted that time. So if you make a clean extraction, your DNA will be able to make long distances, but only in case your DNA is of good quality. In some cases, we have some samples with low quality, and that samples at the day in which make the package, we still look the samples with a very smooth color, and not a clear color. But when the samples arrived at the genotyping institution, they received the samples, these samples with low quality, with a more intense coloration that indicates stated that our samples degraded in the journey, in the travel.
Perfect, Ronald. Thank you so much.
Mm-hmm.
Okay, so we still have 3 hands. So the next one is Marta da Grasa. Please go ahead.
Thank you, Verónica, Ronald, for great presentation. I don't know if you can hear me properly.
Hello, I didn't hear you. Do you
My
have a
question?
question? Okay.
Yes, I do have. Can I go ahead?
Yes, please. We are already beyond time. Please go ahead.
My question is related to the first presentation of Ronald on duplication of accession. So I would like to know if you have insights on how we can properly manage our germoplasm while preparing for the DNA extraction. Most of the time we collect our samples, we deal with this in our gene bank, in which we are getting this from farmers field. So most of the time the samples are mixed. So while bringing them to our gene bank, we used to have duplication while doing the extraction of DNA. So I would like to know from you if you have insight on how we can better avoid the duplication of these samples. Thank you. This is my question.
Sorry, Mary, can you repeat me the question?
Yeah, she wants to know. She said that they do sometimes the collection in the farmer field and then they have the duplicate. How can they avoid duplicate in their collection by using this
During
method
DNA extraction.
during DNA extraction?
How to avoid the duplicate
To
of the
identify
sample?
duplicate, at least to identify them, yeah.
Yes, yes. When I put the examples of the analysis of duplicates, the samples that I used were collected at different times. In some occasions, when the DNA quality is not as good, there is a batch effect in the results. And for example, you will see a group. The samples are grouped by not by the real genetic distances, but by an artificial grouping. In some occasion is by the batch of the extractions or the batch of the processing of the samples. As I understand your question is how we avoid the appearance of these effects in our analysis. Because if you have a real duplicate in your collection, you will be observe and verify this duplicate by the distance of this pair of samples compared to the distance of the entire collection. And in this reason, for that reason, you need to make controls, include controls in your analysis to obtain the distance in which repeated replicate samples are observed. Because always, if you repeat one samples and make another extraction and analyze replicates, you also will find very small, but you will find differences in the genotypes that can be attributed to the DNA quality, but also to the technique. So for any kind of marker you will use, for the duplicates, you need to establish a methodology in which you will genotype your samples, but also include a very large amount of replicates to differentiate in which cases the distance are attributed to real duplicates and in which other cases are attributed to technical effects.
Okay, thank you. So now we have Carla who want to ask the question. Please open your microphone and go ahead, please.
Yes. Hello, can you hear me?
Yes, we can hear you very well.
I have two questions. One is, do you have any insights regarding fresh samples and storing them directly at -80 degrees for a few days or few weeks before we do the extraction? Because we don't have liquid nitrogen, so when we prepare samples, we just take them from the in vitro tubes and we place them in EPIs at -80. Do you think storing them like this for a few weeks can impact the quality, the quality of the DNA, because we are struggling with that? And also, is there a particular reason why you place the samples at 65 degrees for 45 minutes at the beginning of the extraction? Because we usually do like 10 minutes. So yeah, that's my questions. Thank you.
If I understand, your question is that your samples are stored at 8 degrees directly, your fresh tissue, and
-80.
at -80. And after that, you directly from that -80, how would you make the grind of the tissue?
So we store them in EPIs with like plastic beads, and they stay in the freezer for like 1 week or a few days or a few weeks.
Mm-hmm.
And then when we have time to do the extraction, we grind them directly from the, we take them out and we grind them and then we start the extraction.
Yes, if you have these considerations, there is no an effect, or the quality of your samples are not attributed to the condition of your samples. Your samples are well preserved. Maybe you can look another reasons why your DNA is going with low quality, but how you all are working is well.
Yeah,
Thank you.
just to complement, sometimes the ideal is to work the same day. We collect, we extract if we have the opportunity to do it fresh, but sometimes we have that many samples, so many that we have to work many days. So sometimes we collect one day and start starting the rest of the days. So as Ronald said, it's okay that you put it on the freezer, but you can consider using more mercaptoethanol if you're doing setup. If you plan to have it more time, maybe it's better if you if you don't put 2 microliters of mercaptoethanol, maybe try with 4 or even 6 or 8. If for your plant maybe it needs more mercaptoethanol or other type of reagents to conserve better.
Okay. Okay, because we store them with nothing. We just put the fresh leaves in the tubes and then we store them like this.
Oh, you don't put it on the buffer and freeze it with the buffer. Okay. Yeah, but it's okay to, right? You can do yeah. Yeah, but maybe try put it in the on setup with mercaptoethanol and -20 maybe, if you're going to work in 1, 2, 3 days, right?
Thank you.
Okay, then we have the Ama. Please, these are the 3 last questions now because we are already beyond the time. And Ama, please go ahead.
Hello, do you hear me?
Yes, very well.
Yeah. My name is Ronald. I'm from Saudi Arabia. Also, I'm the focal point of Saudi Arabia with FAO. First of all, many thanks for organizing this workshop with the wonderful scientist. My question for Flanka, what is the best approach for extract high quality RNA from the old sample that was not properly reset? I mean, are there any protocol you recommended for degraded materials? Thank you.
Yes, well, we don't normally work with degraded materials, but I know there is specific protocols for this. I think that there are some kits, some commercial kits for old materials, right? But at the end, you will have to deal with low quality, I think. So what do you think, Ronald?
Yeah, great. If you can share me which brand or what's the name for the kits?
Oh, yeah, no, I don't. Well, as I said, we don't normally work with degraded materials. I can look at it, but I don't know if I would find something specific for this type of materials. What type of materials are there? Herbarium?
Yeah, it's about a few years or 10 years ago, but it's not probably received well.
Oh, yeah. Yeah. I don't know. What do you think?
Yes, I want just to say that I remember long ago when I was working on I was doing gene extraction for rice, leaf sample.
Yeah.
So because I was using microsatellite, but people don't use it anymore. When the DNA was degraded, it was still working because you have microsatellite everywhere on the genome. So you can still match somewhere and then amplify. It was a good marker for any environment where you are. So, but now where we move on the SNPs, it's very, now you have to really to have a good quality DNA.
Okay, clear.
Okay, so now it's Mary, please. Mary, you have the floor, please.
Yes, hi. First of all, I would just to thank for all the informative presentations. So I really appreciate how you explain those processes. And this is my question. So I'm actually curious if um what is the minimum DNA yield which you can still utilize and produce results, and is it consistent regardless the crop, or does it depend on the crop? And if one did extracted low DNA yield, does it mean that you restart the process and discard the collected DNA, or does that um, DNA yield still have any of use? So that's my question. Thank you.
She has 3 questions. Please, who wants to take them?
Your microphone is off, Ronald.
Oh, yeah. I'm sorry. Yes, I take the first question. How the minimum amount of DNA you can still consider a good extraction? It depends for the method of the genotyping you will use or the sequencing. And also depends of the, as Mary Noel mentioned, the kind of marker, because, for example, in CTAP method, for our collection of tubers and roots in SIP, we need 50 nanograms per microliter, let's say an amount, and our extraction is good enough to reach these requirements, and are requirements for GBS or sequencing methods that relies on enzyme digestion. But for more delicate, more strict methods like the genome sequencing, maybe our concentration needs to be reevaluated, because the method in which we count the amount of DNA is the agarose gels and the spectrophotometric method. And these 2 methods give us specific values. When you need a more specific marker or sequencing platform, you are required to quantify the double strand DNA. And for that quantification, you need another equipment to verify the amount of DNA. Methods that relies on the fluorescence of the DNA combined with one fluorochrome. And for that reasons, if you obtain even 10 nanograms per microliter, but your DNA is the double strand DNA verified with a high throughput method like spectrophotometric, even these 10 nanograms per microliter are good for downstream applications.
Okay, so thank you very much.
Okay, all the questions were addressed, Mary?
Yes, I just one. So, if you obtain lower DNA yields compared to your target, so do you discard those extractions, or does it still have any of use?
Well, I think that if you have low yield but good quality DNA, you can sometimes you can adapt samples that is the same, the repetition, to go to reach your goal, or I don't know, 30 or 20 nanograms per milliliter, right? So you have 10 that are good, and then you have another 10 in another one. You can lyophilize those together to have a good sample. So it can be useful. But if it's bad quality and bad yield, maybe not that worth it.
Okay, thank you for your answers.
Thank you very much, Mary. Now we give the floor to Valverde. Please, I hope I pronounce well, sorry if not. Valverde, can you hear me?
Marilu Valverde.
If we no answer from him, then we go to the last one. Rimati, please. Can you raise a question? If not, please, we go to Ratemo, please. Then after that, Ratemo, you raise your hand. If you have no question. Okay, I think there is no more question because they are not speaking. So thank you so much. I think we are almost at one. We have 1 hour beyond the time that we were supposed to stop this presentation. So tomorrow we will thank you so much for the participant. Thank you so much for the presenter. Please join me to say thank you to the presenter. So you did a great job today, and tomorrow we will continue with the third session. It will start at 1 p.m. European time, and we will be closed around 3 p.m. European time. Please mark your calendar so that we start on time. When we start on time, we can also stop on time. Thank you so much to Ronald and Veronica. Thank you. Goodbye. See you tomorrow.
Thank
Goodbye.
you. Goodbye.